Hello everyone and welcome to my presentation on our research project on identifying mechanisms of insect repellents and polypore fungi. This project was recently funded by the National Science Foundation, or NSF, and it's going to be conducted at Kean University at the biology department, and we will need some high school students to participate in it and help us with conducting of this exciting research. I am going to start with some general introduction into fungi, what they are, why fungi grow on wood, what kind of fungi grow on wood, what they do in there, and what kind of chemicals can we find in the fungi, and what would these chemicals do, or what would these metabolites, in other words, would do, and what would be their functions. And then we're going to move on with the whole purpose of our research and what we do and why. All right, so fungi is a whole kingdom. That is not plants, that is not animals. Fungi are heterotrophs. That means they absorb nutrients from the outside of their bodies. They cannot perform photosynthesis, such as plants. They cannot make sugars out of carbon dioxide and water using sun energy. They also don't have stomachs, they don't have digestion systems, so they cannot really break down complex organic matter inside their bodies and then digest it and use these smaller nutrients to grow their own bodies. So what they do, they synthesize enzymes, they release them, and the enzymes break down lots of complex molecules around the fungi, and then the fungi absorb all these smaller broken down molecules and use them to provide energy to grow their own bodies. So the fungi actually is a bunch of something that they call a hyphae. Hyphae is a thread. It's a very long thread with multiple nuclei inside. And what you guys see and call a mushroom is pretty much a densely packed body or made of hyphae. A mass of hyphae is called a mycelium. So mycelium can be either diffused or can form this body of a mushroom, and usually this body would serve some kind of reproduction function. It's going to make some spores, and spores will be released outside, and the wind is going to move the spores away, so fungi can grow elsewhere and distribute themselves around the forest or some other area. Okay, so most of the fungi are going to be decomposers. And that means they are going to live on something that's already dead, some kind of plant matter. Most of the plant matter is going to be decomposed by the fungi. You can see this dead wood, cellulose and lignin, all these chemical molecules that are left from the trunk of the tree are going to be broken down by these fungi. And we will never see the huge pack of wood in the forest that stays there forever. At some point, fungi will take care of it and decompose and destroy all of this. Those are decomposing fruits, some kind of oranges in here, I believe, with mold. Those are typical decomposers as well. So parasites would affect the living organisms. This is a parasitic fungus that causes these spots on a plant. This is a parasitic fungus that grows on an insect. And some fungi would form so-called mutualistic relationships with, let's say, plants. This is a lichen. It's a fungus plus cyanobacteria, photosynthetic bacteria. Altogether, they form a lichen that can grow, let's say, on a tree or on a stone or on the soil. And the fungus would provide some nutrients it can absorb from the outside, and the photosynthetic bacterium, cyanobacterium, will do photosynthesis, so they would exchange the nutrients with each other and help each other to live in this symbiotic mutualistic relationship. Or some fungi would appear to be living separately, but actually underneath in the soil, their hyphae connect to the roots of the trees around, and again, both of them would help each other to grow and live in the forest in this symbiotic relationship. So in your life, you probably have seen lots of fungi and fungal products. Of course, the first thing we think about when we talk about fungi is mushrooms that we eat. It's a protein-rich food, so it can often work as a meat substitution for people who do not eat meat. This is Agaricus bisporus. It's a Latin name for a common mushroom that you can easily buy at pretty much any store. So certain fungi would produce drugs such as antibiotics, penicillins, cyclosporins, certain vitamins, maybe anti-cancer chemicals, psychedelic substances such as psilocybin, or even statins, the drugs that lower the cholesterol. So on this plate there is a penicillin that produces the penicillins, antibiotics, but there are lots of fungi that produce those chemicals, those metabolites that we can use as in medicine. From certain fungi you can make something that looks like a textile. It's almost like a leather. And also certain fungi can be genetically modified to produce pharmaceutical products. For example, yeast is a fungus that is very popular in biotechnology, can make lots of things in it. Certain things like insulin are produced biotechnologically. All right, so think for a moment, how many fungal species do you think are there in the world? So we're gonna give it a couple of seconds and then I will reveal the answer. And the answer is more than 3.5 million species. Of course, we don't know every single species. We can't really assess every single one, but this is our estimate based on what we already know. We don't know that many species, maybe several hundred thousands, but everything else is still there out there in the world for you guys to study, and for us as well. Okay, so certain fungi will grow on wood, and those fungi mostly are going to be decomposers, and you can see how beautiful they are. All these guys grow here in New Jersey. They can be small, they can be large, they can be edible, some of them, and the wood fungi would possess very specific enzymes that work as special tools to break lignin and cellulose, the components that actually build up the wood. This wood is very, very hard. Bacteria cannot really break it down, with very, very small exceptions. So most of this wood breaking is going to be conducted by the fungi. So fungi in the forest are going to be the most important decomposers to break down dead wood. There are also parasitic fungi that infect the living trees. And we're gonna see that some of them are also very interested in terms of their physiology and lifestyle. All right, so let's take a look. What's the difference between these two forest pictures? See number 1 and number 2. So you can think for a second, and you can probably see that on this one we have a lot of dead wood, right? And on this one we don't have any dead wood. So this is how a normal wild forest would look like. All these trees at some point were dead, and then now they're fallen trunks, and you can see this one is pretty fresh. and this one has already some moss growing on it, and eventually all of that is going to be decomposed, and minerals will go to the soil, and the carbon components will convert into CO2 in the metabolism of fungi and will be released back to the air. Right, and this picture was taken by me in Weyanda Park here in Jersey. And you can see how clean the park is. It's because the park is heavily managed, and this dead wood is constantly removed to make the trails nicer for the tourists. So you can imagine that fungal species that grow on wood and decompose the wood, they are present in this forest and they are absent in this forest. So when we're talking about the healthy ecosystems that include as many species of all different organisms as possible, we have to also think on the microorganisms such as fungi that must be a part of the healthy forest. So they must grow in that forest, and if we remove their normal substrate, their normal food, then we're going to lose them as well. And some of them can even become extinct. It's not good for a forest, it's not good for us, because many of the wood fungi are super interested in their metabolism, and we can learn something from it, which is a goal of our research project. All right, so famous wood fungi that grow on wood, on a wood that is dead, that are not parasitic, okay? Well, not necessarily. Some of them are like half parasites. They start growing on the live wood, and then eventually the tree dies and they continue growing on it as well. So Ganoderma lucidum, or reishi, you've heard about reishi. It's a component of lots of kind of mushroom coffees. We believe it contains lots of metabolites, chemicals that help mental state of humans, that help with the overall health, reduce chances for people to develop cancer. So it's a supplement that you can often find in different mushroom mixtures that are sold in the pharmacies. So hen of the wood, chicken of the wood, those are edible mushrooms that we can collect and actually cook and eat them. Turkey tail has the same kind of famous name as reishi, or Philinus linteus, which is San Juan, kind of also famous in China. So Tremendous versicolous, turkey tail, and San Juan and reishi, they all are famous for their medicinal properties, and their components are often used in different mixtures and supplements, including coffee supplements, that are promoted as being healthy for human consumption. All right, so what is the role of wood fungi besides pure decomposition, decaying of dead wood? Besides the fact that dead wood should actually disappear and should go somewhere, right? What else can be a use in the forest of a dead wood? Well, this dead wood is used by lots of other organisms. You've seen the moss growing on the lying log, fallen trunk of a tree previously on a previous slide. So lots of plants would use the dead wood that accumulate a lot of water in it as a substrate to grow on. And insects would live in that dead wood, and larger animals such as birds would live in the dead wood as well. For example, woodpeckers cannot live in the forest that doesn't have a standing dead wood. And where is this kind of dead wood coming from? It's coming from the fungi that grow on this wood, right? They kind of facilitate rotting of the wood inside, and then it's much easier for the woodpecker to kind of make those nests, so the woodpecker can live inside. And actually, woodpeckers move the spores of these wood fungi from one dying tree to another. They come to the tree with like certain cracks. They start developing the nest, and then they leave. But the spores from their beaks get inside and continue to work, and now the tree is going to become softer, and the inside is going to die. The tree is going to become hollow. The woodpecker can get back and develop the nest even more. You can see here, this is the growing fruiting body of the wood mushroom, and this is a nest where the woodpecker is going to live. And you can see it's a whole hotel for the woodpeckers. Several woodpeckers made these nests, and they're going to live in there. So woodpeckers actually cannot live without those nests, and if the forest is very clean, no standing dead wood, it's kind of all fresh and clean, then the woodpeckers won't have any place to hide at night, and they're all going to be pretty much eliminated by night predators, such as, for example, owls. So the woodpecker and wood decaying fungi form a very important mutualistic relationship. The relationship that results in benefits for both of them. Okay, so fungi form the relationships with insects. Insects are much smaller than woodpeckers, but they're also very important for the ecosystem of the forest. And the very small ones are going to live in those wood fungi. They're going to make little houses in there, and they're going to eat the flesh of the fungus, and overall they're going to live on it. So different species of wood fungi are pretty famous for the formation of the nice, good, beneficial relationship with the insects as well. All right, so it's actually not very easy to study the wood fungi that grow on wood. In most cases, this is what they're going to deal with. This is the mycelium and combination of hyphae from the fungus you're working with, so you can't really say what it is. You would have to analyze DNA, and I'm going to explain just a little bit on the next slide. So this is actually something high school students can help me with. They can help with the analysis of the DNA and identification of the fungal species, what exactly we are working with and dealing with. Many different species of deadwood fungi would have very similar fruiting bodies. So imagine that this mycelium actually packed and grew into a fruiting body. So this is Philinus igniarius and this is Philinus tremulae. Those are Latin names for the species we work with, and they look very similar. If you're not a specialist, you may actually make a mistake and think that one is one and the other one is the other. Again, we would need the DNA analysis to confirm what they are. And on the top of that, depending on how decomposed the tree is, different species can grow in there. And let's say I want to look at the Philinus tremulae, but it grows on a tree of a certain species at a certain condition, so I wouldn't be able to find it until I find this tree. It's not like it's growing everywhere, so that's an addition to the challenge in studying of the fungi. All right, so I promised you to explain how the DNA analysis works. So basically we have to isolate the DNA from the fungal sample and then find the region in there that's called ITS, internal transcribed sequence. This region is like a fingerprint for the fungus. We can study it with certain methods in the laboratory, identify the sequence of nucleotides in the DNA of ITS, use the reference database to identify the species using this ITS gene. Same idea with the fingerprints. If you know the fingerprint and you have a reference sequence in a database, you can compare, you know who the person is. Same for the fungal species and their ITS genes. We don't need the whole genome of the fungus. We just need a very small gene to actually confirm that this is a species we work with. Okay, so high school students actually perform very, very well and helping me with this process a lot. And if you guys decide to enroll in this project, you will work with DNA isolation, purification of DNA, polymerase chain reaction. This is to make a lot of copies of ITS gene to study it. Electrophoresis, DNA sequence analysis, all of these are molecular biology techniques that you're gonna guys do in the lab using all these pipettes and different solutions in different instruments to actually see what the sequence of the gene is and what the species is based on the sequence analysis. Okay, I have worked with a lot of undergraduate students before. Some of them made it into publications. They became co-authors of papers that we published from my lab. These guys are in red. I haven't published with a high school student yet, but if you are a student who want to spend a lot of time on the research, chances are you will be able to make it to a co-author of a paper as well. And of course, at the end of a project, you will prepare a poster presentation for your symposium, or maybe we can even take it to some other high school science conferences, and you would be able to present it outside of your school as well. All right, so a little bit about the project, specific project I'm working with right now. We're working with a fungus that's called Felinus tremulae. These are the fruiting bodies, the compact mycelium structures that are growing on aspens. Populus tremulae. Okay, this is the trembling aspen. This fungus, Felinus tremulae, or aspen bracket, wants to grow only on Populus tremulae. Very rare on big tooth aspen, which is a Populus granifolia, and we'll still have to confirm on why it wants to grow on some other trees as well. But basically, it's a parasite of the trembling aspen, wants to grow only on this tree. What does it mean? It does need certain chemicals, certain metabolites from the tree to work on, and there is some kind of relationship, chemical relationship between these two guys, the tree and the fungus, that is very interesting for us to look at. The thing is that this fungal species doesn't form any relationships with insects whatsoever. All entomologists that work on insects, that work on insects that live on the deadwood fungi, say we've never seen anything on this fungi. What does it mean for us? For us, it means chances are there are some kind of chemicals in this fungus that prevent insects from living in there, and potentially these chemicals can become insecticides that we can use in our live naturally derived insecticides. So we're definitely not at the stage when we can develop an insecticide. What we can do is we can study the chemicals inside the fungus and see what they are, maybe test them on pest insects first and see if our hypothesis that the aspen bracket actually has some insecticide inside is correct. All right, in order to study the chemicals, we have an instrument that's called liquid chromatography mass spectrometer to perform this spectrometry analysis. It's a huge instrument, takes the whole room. So what we do, we smash the tissues of the fungi, extract some chemicals and metabolites from it, and then analyze using this instrument. And then the instrument, of course, is going to give us something that we can't read at first. So there's some kind of computer analysis. The instrument is basically a very, very sensitive scale. So it will measure the weight of all molecules in the mixture. We will use a chromatography technique to kind of separate those chemicals into smaller groups so we can look at them in more details. And then the spectrometer will tell us what's the mass of all of these chemicals, molecular weight, basically. And then it will also tell how much of everything we have with a specific molecular weight. And after that, we'll just have to deduce what are we dealing with. This also requires a certain reference database, means we will compare all these fragments, chemical molecules, and their little parts and their molecular weights to what we already have in the reference database, and maybe we can even figure out what it is and name it somehow for the compounds that nobody have ever seen before, and it's a pretty likely scenario. We would have to then just guess the number of, like, let's say, carbons or oxygens or hydrogens in it, but that's basically what we can do. For our purpose, it's not as important, actually, to know what exactly this molecule is. If this is a molecule I can separate from the mixture and test on the insect and see if the insect is sensitive to it or not, that's already good, because there are other methods that will look into our molecules even further, and then we will see what they are, but not at this stage. All right, so what we already did, we collected different Philinus species, some from Jersey, some from other states, looked at the metabolites inside, checked what metabolites are specific to our fungus Philinus tremulae, the one that we're really interested in. Found some very, very special metabolites inside that we cannot find in other very similar fungi that actually have some relationship with insects. So these specific ones are probably the reason why our fungus repels the insects. And we just need to do some more statistics. We need to do some more of a testing. I'll look a bit more in details on what these molecules are, and we will already have some fundamental information, fundamental knowledge on what our fungus can make that makes it insecticidal. And of course, you guys can help with the DNA confirmation of each species, what exactly we're working with, and you can help with extraction of metabolites and preparation samples for analysis. Okay, and the testing on insects is going to be done at Rutgers University, but first we have to prepare all the samples. We're working on it. We're going to also see if the concentration of those chemicals is important. We want to know if the fungus makes these molecules by itself, or is it accumulating those molecules from the tree. That's a good question. So in order to test this hypothesis, we're going to try to grow some fungi in the lab. We have a growth chamber for the fungi on the way, so we'll play with some culturing of fungi in the lab. And we will see if my fungus can synthesize those chemicals from scratch, or you have to provide the tree for this chemistry inside. All right, so in summary, you guys will help with the DNA barcoding analysis of fungal species using the DNA. You will help with extractions of metabolites, isolation of certain components, and you can definitely help with growing fungi in the lab and compare how they look like if they grow on a parasitic tree or they grow without the parasitic tree, because that would give us an idea on what exactly is synthesized by the fungus by itself and what exactly is absorbed from the tree. Okay, so we're looking for high school students for the spring of 26, for the spring of 27, and for the spring of 28. So it's 6 students per spring. Okay, so for the upcoming semester, I would need 6 students. These students can work with me in the lab, or they can work with Professor Carr, who will provide a separate presentation for you. Professor Carr is a computational chemist, so he will work more with the computational analysis of the molecules we're working with, and we guys are going to work more with the lab and pipettes and actual cultures. So you can select if you want to work with me or Professor Carr, and you will be provided a stipend. We'll have to do some paperwork for that, but this is a stipend from the National Science Foundation, and you will definitely be paid for your work during your spring semester, and you will be able to present your work at your symposia in your high school or maybe some other conferences. All right, that's it, guys. I hope this was useful for you, and see you in the lab.